McElhaney, and today we are talking about plateable hepatocytes. We get a lot of questions from clients, and today we're going to discuss a few of these. Plateable hepatocytes have an array of uses. One common use is for induction studies. However, there are many other applications as well. You can use plateable hepatocytes to look at metabolism, biochemistry, virology, and cell biology. These are not just for drug development. All hepatocyte lots undergo rigorous testing to look for attachment characteristics. Once those characteristics are determined, we then further test to optimize for seeding density and to provide our clients with mRNA and enzymatic induction data. To be qualified as attaching, cells must form a monolayer that is at least 85% confluent and also last through a 3-day induction assay. These cultures generally will survive much longer in culture. At XenoTech, we are here to help, and we can provide technical support and help you pick which test system is best for you. Animal hepatocytes from small common laboratory animals generally have around the same activity, with the exception being outbreed species like XenoTech. Human donors differ greatly, so choosing the right donor is something that we can assist with. This is a benefit of our CryostaX product, because this product can be customized to fit your needs. Additional benefits of the CryostaX format is the quick turnaround time. These are a single freeze cooling method, so we are able to skip additional steps and time that are associated with traditional cooling methods. At XenoTech, we test each lot to optimize seeding density before it is ever released for client use. Each product has an assured minimum yield. This is our guaranteed number of cells per vial, and we do numerous test calls to ensure that each lot meets that yield. All of this data is included on our product data sheet. You can use these numbers to determine how many vials you will require for your needs. Seeding density is broken up on the data sheet by well format. 48 well and 96 well plates are a little more difficult to culture because of their small well size. This number has been accommodated for and adjusted and is available on the data sheet as well. When seeding, the speed should be slower so that the cells are gently added to the wells. Seeding too quickly can cause gaps in confluency and disruptions in the collagen coating. The only difference between CryostaX vials and traditional vials is the method of which they are cryopreserved. CryostaX vials are cryopreserved in pellet form. This is very important because it eliminates the need for a water bath. A very common mistake when thawing hepatocyte is keeping your cells in the water bath for too long. This can greatly impact your yield and viability. So by eliminating this step, this improves your chances of having a successful thaw. CryostaX can also be completely customized to suit your needs. We can customize donors, yield, individuals, and pools quickly and easily without custom product pricing. Cell counts are essential for successful cultures. Both overseeding and underseeding can lead to underconfluent cultures. So being consistent with your technique can help ensure you have the most accurate counts possible. Hepatocytes settle very quickly. The cells should be resuspended thoroughly, ensuring they are completely mixed before removing your cell count sample. Although your cells should never be vortex, it's very important to keep your cells resuspended throughout the process. The cell count sample should be taken from around the middle of the volume of your cell suspension. This practice ensures you will not get a pocket of cells included in your count. This would elevate your yield. When training, I always suggest bringing your pipette tip all the way down to the bottom of the tube and bringing it up slightly to take your sample from around the middle. When adding your cell suspension to your counting solution, the pipette tip should barely touch the tip of your surface. This is so that cells on the outside of the tip are not included in your cell count. Do not pipette up and down to mix your sample, as this can shear your hepatocytes and can lower your viability. Instead, gently rock your sample back and forth to mix your sample before adding it to the hemocytometer. It's a good rule of thumb to have your cells on a plate within 30 minutes of thawing. It is helpful to set up everything you may need, label your plates and tubes before you ever remove your vial from cryo storage. We also recommend that all media is warmed to 37 degrees Celsius prior to beginning any procedure. However, it is not necessary to keep your cells and media in the warmer throughout your counting process, and room temperature conditions are fine. While most species have an overlay period of 2 to 4 hours, rat hepatocytes have a delayed overlay time. This is because rat hepatocytes have a natural gapping pattern that can make your cultures appear under confluent. Waiting until the next day to overlay gives the cells a greater opportunity to flatten and form a confluent monolayer. Our OptiMatrix overlay is recommended because when you culture cells in a sandwich culture, the cells regain polarity, form canalicular networks, and retain more in vivo-like properties. Hepatocytes also retain their morphology throughout the culture period. 96-well cultures are a very tricky format to work with. This is because well-to-well variability is a common issue. We recommend using the reverse pipetting method. This means going down all the way to the second stop before pulling up any cell suspension. When dispensing your cells, go down only to the first stop. We do this because it eliminates the possibility that air bubbles will be introduced into your wells. Air bubbles can disrupt monolayer formation, and in 96-well plates, that can make entire wells unusable. We also recommend using manual multi-channel pipettes. If electronic multi-channels are used, make sure the speed is turned down and only dispense half the plate at a time. This is because cells can settle in the tips, and this leads to uneven distribution of cells in the wells. The use of repeaters should be reserved for larger well formats, again set at a lower speed. Manual pipettes are recommended for 96-well plates. We hope you found this helpful, so thank you for watching.